Hey guys, it's Professor Winkleman here. I'm going to make a podcast for Thursday's lecture. So we're going to cover blood vessels, blood itself, and circulation, and we're going to go into the arteries, veins of the lower body. Hoping that this will allow us to have activity time in class and discussion time over this topic, and this will also provide you a nice resource to go back to listen to over the material. So please take some time to watch this video. So first we're going to talk about the functions of blood. So as you know, blood makes up a large amount of our body. Inside blood, we have a whole bunch of different structures which we'll get into specifically for the anatomy of blood. But overall, the functions of blood are transportation of oxygen and carbon dioxide as well as nutrients and waste products. When this all happens, it's going to be able to help us regulate our body temperature, our pH, and our fluid volume. It's also going to protect by mounting an immune response and production of antibodies. And we'll get back to some of these later throughout the presentation. If you think about it, when we talk about blood and what the blood cells are going to do, it's going to transport oxygen and carbon dioxide. It's going to take that throughout your whole body, through the heart and through your lungs, and then send it to where it needs to go afterwards. It's going to be able to regulate your body temperature. So when you get cold, your blood is going to attempt to regulate that. Or if you get too hot, it's going to attempt to regulate that as well. And we'll get into pH. Remember, we talk about 0 to 7 and then 7 to 14 for pH. What we're looking at here is a picture of composition of blood. So on the first picture on the left, you're seeing a syringe being injected into an individual's elbow into withdraw some blood. From this, it's going to enter a centrifuge. That is the picture in the middle. The vial of blood, as you can see, taken straight out of the arm is whole blood. Once it's put into the vial and then that's inserted into a centrifuge, it's going to spin that vial or that test tube of blood. Once it's done spinning and you take that vial out, that's what you're seeing right here on the final step 3. As you can tell, we still have what our red blood you would normally think about, and that's our erythrocytes, and that makes about 44% of whole blood. The other 55% is plasma, and that's the top portion. You got a 1% or almost less than 1% of a buffy coat of leukocytes and platelets. Okay, so we have some specific elements that are formed that make up the components of blood separated post-centrifugation. As we talked about, we have the composition of blood. After we go through the centrifugation, blood has 3 main fractions: erythrocytes, a buffy coat, and plasma. If you want to look at each of these individually from this diagram, the top part is going to be the plasma, and we see that this distinct characteristics of plasma are water. So out of this 55% of plasma, 92% of that is water. And then we have some proteins, about 7%. So we have albumins, globulins, fibrinogen, and regulatory proteins. And then other solutes. So we have some electrolytes, some nutrients, respiratory gases, and waste products. I'd like to note that 92% of plasma in this blood is water. So if you think about over half of your blood is plasma, and almost all of that plasma is water, you have a lot of water running through your body, right? So the composition of blood really is dictated a lot by the water in your body. The bottom part we talked about with the erythrocytes, you have 4.2 to 6.2 million erythrocytes per cubic millimeter. It's a ton of red blood cells floating around, but it only makes up about 44% of your blood. And then that last one, less than 1% blood, that buffy coat, we have platelets and leukocytes. You can see the different types of leukocytes there: neutrophils, monocytes, eosinophils, basophils, and lymphocytes. Basically, all those are going to be active fighters in the immune system response. So we'll talk about these 3 elements again. So as we talked about on the screens before, we have the formed elements that occurs post-centrifugation. So erythrocytes make up more than 99% of formed elements, and their primary function is transport respiratory gases. Please remember, respiratory gases are carbon dioxide and our oxygen. Leukocytes is the other formed element, and these make up less than 0.1%. And as I said before, these are your immune response in defending bodies against pathogens or disease and infiltrators. So these were going to be like your eosinophils and your basophils. Your platelets make up less than 1% of formed elements, and their primary function is blood clotting. So platelets are kind of like the sticky things that stick together within that formed element to occur for a blood clot to occur. Inside the formed elements, we also have what's called hematocrit. Hematocrit is the percentage of the volume of all formed elements in one's blood. And it varies depending on your gender. So females have about 38 to 46%, and males between 42 to 56% is hematocrit. And this also differs at altitude. So think about the hematocrit again is the percentage of volume of all formed elements in one's blood. So I'm going to go back a screen. So if the percentage of volume of erythrocytes, leukocytes, and platelets within one's blood. Okay, remember that this is not going to be all the aspects of blood, but these are the formed elements of blood. So we got to ask, why do you think this differs at altitude? So think about the Mile High Stadium where the Denver Broncos play, or when you go hiking or skiing up in the Alps. Think about what happens to you. You have a lack of oxygen, and basically it's putting your body into a strenuous environment that changes the normal function. So if we go back another screen, remember erythrocytes make up more than 99% of formed elements. Those are responsible for transporting respiratory gases. If we move into these different altitudes, that change in altitude basically changes the amount of erythrocytes that we're going to be able to have within our formed elements, thus reducing the ability to transport oxygen and carbon dioxide effectively within our body. So this is a picture of an erythrocyte. So erythrocyte is just a red blood cell. So you'll see red blood cell abbreviated RBC. That's a medical abbreviation for red blood cell. Erythrocytes are responsible for transporting oxygen and carbon dioxide to and from tissue. They, as I said, we refer to them as red blood cells, but it's actually a misnomer or a miscommunication because as mature red blood cells, they actually lack a nuclei, whereas erythrocytes in itself, when they are just a red blood cell, have a nucleus. They are relatively small, as you can see, and you have a unique biconcave shape. What that means is bi means two, concave, basically it's going to create this concave on either side. So it's basically going to be an indentation on either side. So kind of think of it like a, I don't know, what it kind of looks like a pizza, if you will, where the outside's of the crust, but it's like a double pizza that you get a top crust and a bottom crust with a thin layer in the middle, creating a biconcave shape. As things pass through small blood vessels, they line up in a single file called a arula. Now if we look at hemoglobin and erythrocytes, every erythrocyte contains 280 million molecules of a red pigmented protein called hemoglobin. So hemoglobin is going to be critical in making up what that red, I guess, picture, that red pigment, if you will, of what erythrocytes look like. Hemoglobin is also capable of reversibly transporting oxygen and carbon dioxide in the blood. So as we just said, erythrocytes are responsible for that. Hemoglobin is capable of reversibly transporting, so back and forth transport. Hemoglobin consists of 4 globin proteins. Globin proteins. We have 2 alpha chains and 2 beta chains. And make sure to know those Greek letters, alpha and beta. Each of the 4 globins possess a non-protein heme group containing of an iron, and that's the chemical symbol for iron, Fe, and it has a chemical iron molecule inside of it. Each hemoglobin molecule can bind a combination of 4 oxygen and carbon dioxide molecules. So each of these 280 million molecules, every erythrocyte has 280 million molecules of hemoglobin inside of it. This hemoglobin then can bind to a combination of 4 oxygen and carbon dioxide molecules. So if you do the math, that comes out to a lot, just under 1 billion ways to transport oxygen and carbon dioxide within the blood. So this is our structure. So remember, we have A is alpha and B is beta. So we have our alpha 2 chain here, alpha 1 chain here, beta 1 chain here, beta 2 chain here. Remember, these are going to have the ability to transport oxygen and respiratory gases, whether that be carbon dioxide or oxygen. The little red structures you see are what we call the hemi. They're a ringed molecule with iron. So remember, anytime we talk about hemi, they have the iron involved in the center of that. So we just finished talking about erythrocytes. Those were our red blood cells. Our leukocytes are our white blood cells, our WBCs. Unlike the erythrocytes, leukocytes possess a nucleus and organelles, and that makes them a true cell. Remember, we talked about a mature red blood cell lacks a nucleus, so that's not actually a true cell. Well, leukocytes do have a nucleus, making them a true cell. These are going to be very critical in your immune response, okay? So they're going to defend against pathogens. So anytime that you get a scrape, a cut, or something hits your arm, your white blood cells are going to be critical in the defense of that pathogen from taking over your immune system. So these white blood cells are going to be the fighters on the front lines, basically, after something has got past your skin and now is into your bloodstream. They are 1.5 to 3 times larger than erythrocytes. Remember, we talked about there's a lot of erythrocytes in the blood, yet they're kind of small. These are a lot larger. White blood cells reside mostly in tissues and not actually in the blood. They are capable of leaving blood vessels called diapedesis and entering a tissue. So that's a good term to know, diapedesis. So they're basically able to exit that blood vessel and then enter the tissue, and they will hang out there. Because if you think about it, some of these pathogens or diseases that will enter typically will go through the skin and then the tissue first before actually getting into a blood vessel or the bloodstream. So it's critical to have these white blood cells in the tissue as well. They're attracted to a site of infection by molecules from damaged cells or invading pathogens. This attraction is called chemotaxis. Okay, so what this is happening is that whenever a pathogen or infection is in the body, the leukocytes will leave the blood vessels and enter the tissue, or leave the tissue and enter the blood vessels wherever they are called to do. So think about this kind of like the military. The white blood cells are going to get a response from the president, or the military will get a response from the president saying, we need to go to wherever that may be to at that site to invade that area and prevent that from further invasion. Okay, so the same thing is going to happen here. So leukocytes are going to go to the site of infection from damaged cells and then help protect from that invading pathogen, and that's called chemotaxis. All right, remember we talked about platelets and how they only make up about 1% of our formed elements, and they're going to be critical when we have blood clots. Blood clotting has a negative connotation because blood clots can break off and cause serious issues within the body. Sometimes blood clotting is actually critical. There's actually a disease where people have the inability to blood clot, and I highly suggest you look that up to see what that is and try to understand the struggles of that. Maybe we'll talk about that Thursday in our discussion. So when we talk about blood clots, we look at 3 main areas. We have erythrocytes, which are red blood cells, our platelets, and then our fibrin. So remember we talked about fibrin is kind of the wiry matrix that makes up a blood clot. The platelets are going to be the things that kind of stick and hold everything together, and the erythrocytes are going to be the things that are going to help us transport oxygen and carbon dioxide. So when we talk about blood, now we've talked about what makes it up and all the formed elements in it, we need to actually talk about where they are housed. So the anatomy of a blood vessel. So think about a vessel just like a fishing vessel or any type of a what's it called? What's it called? I don't remember. Anyways, so the anatomy of blood vessels, so they are going to have the 3 classes of blood vessels, and they're arteries, capillaries, and veins. Each of these are going to have a very different structure, but basically like a fishing vessel they're all going to look a little bit different, but have one main job, and that's going to be house something and transport something. Okay, so the arteries are going to be in charge of transporting blood away from the heart and become progressively smaller as they branch and finally result in capillaries. So when we have the heart, it's basically going to branch off the heart, and the arteries are going to go away. So anytime you see artery, A, away, away, A. So arteries are away from the heart, so they're going to take oxygenated blood away from the heart, and then they're going to get smaller into capillaries throughout the body. Veins return blood to the heart and become progressively larger as they merge closer to the heart. So the veins are going to run really small down in your lower extremity or in your fingers and arms, but once they get back to the heart, the veins are actually much larger. There's three main layers that make up a blood vessel, and they're called your tunics. So you have your tunica intima, tunica media, and tunica externa. So let's break down the root words a little bit so this can make more sense. Tunica intima, intima means intimate or close, and so this is going to be the most internal or close layer here. So that's the tunica intima, is that pink, that light pink area. The tunica media is going to be this larger structure where a lot of our fibrin will lay down. And then tunica externa is going to be an outside barrier, okay? So it's going to be the blue on this one on the right side, and then in the dark red on this side. So you're looking at a artery and a vein here. So anytime we talk about arteries, they're going to be labeled as red. When we talk about veins, they're going to be labeled as blue. And we'll talk about why that is as we go a little bit farther throughout this. A capillary bed is created in the middle of these. So remember, when we talk about arteries, they start off big, get smaller, and then end at a capillary bed, which is pretty small. Where veins are pretty small and they get larger once we get back to that area. The inside of an artery and a vein is called the lumen. The lumen is a very important area. Basically, that's inside of the capillaries, arteries, and veins, and that lumen is going to be the tunnel or the transportation center for all the blood to move inside the vessel. So we look at the difference between an artery and a vein. Arteries are going to be on our right side, they're the red, and veins are going to be the blue. So when we talk about the blood, it's going to leave the heart and away from the heart via arteries. And once it does that, it's going to travel down your descending aorta from your heart, and then you can kind of see some different examples. So we have our tunica intima, media is the larger portion, and externa is the thin layer on the outside. And this is an elastic artery. Here we have a muscular artery, and that's going to have the same thing. So tunica intima, tunica media, and tunica externa. And finally, we have arterioles, or our small arteries, and they have the same portions, tunica intima, media, and externa. We look at a capillary, we have endothelium, or the outside covering, or the inside covering, sorry, in the basement membrane. And then we look at the same thing. We have a venule, that's the smallest vein, a medium-sized vein, and a large vein. So they don't have specific names, but they still have the same areas called intima, media, and externa. So we compare our arteries and veins off this table. We see the lumen diameter in arteries, so basically the middle center part, so this portion here that my mouse is at, is narrower than a vein in an artery and wider in veins. So narrow in artery, wide in veins. The thickness of the wall is thicker in arteries and thinner in veins. If we look for blood pressure, arteries typically have higher than in veins, so larger arteries typically have more than 90 millimeters of mercury, whereas lower in arteries approximately 2 millimeters of mercury. Arteries are responsible for transporting blood away from the heart, while veins transport blood to the heart. So then we talk about the specific type of arteries. So on that last figure, there was three types: elastic, muscular, and arterioles, and these are listed in order. Elastic arteries are the largest of all arteries. These include things like your aorta, your pulmonary, brachiocephalic, common carotids, and subclavians. All of this we will cover again in like week 15 or 16 when we cover the heart. But basically the aorta is going to be near the heart, pulmonary is going to be near your lungs, brachio refers to your arm, and cephalic means your head, so up near your arm and your head. Common carotid is in your neck, and subclavian is below your clavicle. As I said, most examples are near the heart. And these are going to be the large ones that, remember, the arteries start large and get small. They're also called conducting arteries as they conduct blood away from the heart. So these are the main hubs to everything else. So think about this like a train station. The actual Grand Central Station is going to be your heart. Once you start those trains away, they're large and they got a lot of people on board, and they're going to conduct all that blood flow away from the heart. As you get into smaller towns and little small villages, less people are going to be on board, and those trains are going to be smaller in terms of passengers and that blood flow that's needed down to the certain areas. These elastic fibers present in all three tunic allow these arteries to stretch under increased pressure generated blood flow. So elastic arteries are going to be very, pliable, if you will. They're going to also branch into muscular. So arteries from the heart go to elastic arteries and then branch into muscular. Muscular arteries are medium diameter, and they're called the distributing arteries. So they're going to distribute blood to organs and tissues. So once you get to your destination on the train, it's basically going to drop you off and send that blood to organs or tissues, or send you back to different parts in town. Elastic fibers are not found in the tunics, but in two rings located between the three tunics. Muscular arteries have a proportionally thicker tunica media. So the tunica intima was the inside, tunica media was the middle portion, and the tunica externa was the outer. The muscular arteries are going to have a proportionally thicker tunica media just because of the transportation for distribution. Finally, we look at the arterioles. Arterioles are the smallest of the arteries, and they have less than 6 layers of smooth muscle in their whole entire tunica media, so that's going to be very thin. They have sympathetic innervation. So remember, we have sympathetic and parasympathetic when we refer to the nervous system. If you need to go back, this was week 2 or 3 that we talked about sympathetic and parasympathetic. So refer back to the nervous system. So it has a sympathetic innervation to the muscle fibers of the tunica media, which causes vasoconstriction, resulting in elevation of blood pressure. The term vasoconstriction and vasodilation are going to be critical to understanding the flow of blood. So vasal refers to a vessel. Constriction means to get smaller. So this is going to make your vessels get smaller, resulting in elevated blood pressure. That doesn't make sense to you as you listen. Basically, if you take something that is this shape as the O, pretend like this is your vessel, and you make it smaller on the sides, it's going to make that blood harder to get through there, thus increasing the pressure for it to move. So you have an elevation of blood pressure because it's harder for it to move because it's a smaller vessel. Parasympathetic innervation causes vasodilation and a lowering of blood pressure. So basically, that's going to be the expansion of the outside walls or the inner tunica for the vessel to dilate or get larger, which lowers the blood pressure as it's able to easier move along. So then we look at capillaries. These are the smallest of all blood vessels, and their diameter is only slightly larger than an erythrocyte. So think about a red blood cell and how small it was, and the capillary is the smallest of the blood vessels, and it's only big enough, just big enough to allow one erythrocyte down. The wall consists solely of the tunica intima, a single layer of endothelial cells. So they're very thin and easily broken, if you will. Only type of blood vessel where metabolic exchange can occur between blood and cells outside of the bloodstream. So in your capillaries, you're able to share metabolic exchange, which is going to be critical in the injury healing process, as you'll continue to learn throughout your health care profession. The capillaries will form capillary beds. So a single capillary grouped together with a whole bunch of capillaries creates a capillary bed. The capillary bed structure, so you'll see you have an arteriole, and off that arteriole you see the meta-arteriole and the thoroughfare channels in between. Basically, this is going to create a bed structure here. You see the endothelium is the thin wall of the vein at the bottom. Basically, what's going to create is an arterial end at the top, and it's going to travel through a mix of capillaries in the middle. And these are going to be the true capillaries, and then it's going to go into the venous side. So basically, this is going to be the capillary bed, or the exchange section that it occurs for it to go from oxygenated blood to deoxygenated blood. Veins are blood vessels that drain capillaries and return blood to the heart. And the pressure in veins is much lower than arteries, and at rest, the body's veins hold about 60% of the body's blood. Thus, veins function as blood reservoirs. So as we talked about arteries, they're going to be in charge of distribution and movement, where veins are going to be more of our reservoir. The first of the smallest, so remember last time we started the largest, so we're making a round circle here. So venules are the smallest of the veins, and this is where we start, so they are going to be the smallest to the largest. They're companion with the arterioles, they're at the same level, and basically the smallest ones are located at the distal end of a capillary bed and called postcapillary venules. So if we go back to this, it's going to be this area down here, the postcapillary venule. We all have diapedesis, the migration of leukocytes from the bloodstream to the body, and this occurs through the walls of the postcapillary venules because these walls are so thin. Next we have veins, and veins are larger than venules, but they are smaller and medium-sized veins travel with muscular arteries. So remember, muscular were our medium-sized, so we have medium-sized veins for those. Where large veins travel with elastic arteries, so we talk about our elastic ones near the heart, up near the head, and those are our large veins where we need that blood near our heart and our brain. Blood pressure in veins is too low to overcome the forces of gravity and possesses valves. So in veins, it's not all gravity dependent, and you have a valve that will open and close, creating a blood pressure regulation. Valves are formed from the tunica intima and prevent blood from pooling in the limbs. So they're going to be critical that your valves function properly. When we have pooling of blood in the limbs, you get things like deep vein thrombosis, blood clots, and some other basic critical pathologies that can result in negatory effects. In addition, as skeletal muscles contract, they also pump blood toward the heart. This process is called the skeletal muscle pump. So here what you're looking at is the skeletal muscle pump. So as these two muscles on either side, as this muscle here and this muscle contract, basically they're going to push blood from the tissues when it contracts, increasing the pressure, opening the valve, and then it's going to send blood up to the heart. So as muscles contract, they also pump blood toward the heart. Alright, so if we look here, this is our blood pressure. So typically you think about blood pressure, you put the cuff around your arm, the doctor listens, and gives you a number. So 120/80 is what we've all been told. That should be somewhere around there. So you have your systolic and your diastolic. So your top number is your systolic, and that should be around 120. Active, healthy individual. For the students in my class, you typically are between 18 to 25, so you should be around 110 area. Diastolic pressure is about the 80 range. If you're active and healthy and young, it could be about in the 70s. So 110 to 110/70 is a good blood pressure for you guys. When we look at the heart, we see that these numbers are a little bit more elevated because the arteries are larger here. When we keep going down to a normal artery, we get the same, about 110/70. And then as we get smaller to the veins, the venules, and the capillaries, we basically are not able to record a pressure. Remember, we talked about the veins and the venules not having the ability to a pressure gradient from gravity, and has to rely on our valves to do so. Alright, so take a little break, pause the video, and come back. So we're going to talk about the nervous and vascular system of the lower extremity, and this is part 2. Welcome back. So the vascular system is going to be our first part. When we talk about the vascular system, these are arteries flowing through the leg. So this is going to be arteries, remember, away from the heart, and this is going to be the first part. The main area for blood flow through the lower leg is going to come off the common iliac artery and split into the external right and left iliac arteries. So common iliac is going to be near the iliac crest or the hip, top of the hip bone, and split off to the external and internal, or external right and left iliac arteries. This artery is going to supply blood to the entire lower limb, and the name is going to change as it travels along. So if we follow this artery down, so we're following this iliac artery where my mouse is, basically the name is going to change to the femoral artery as it passes inferior to the inguinal ligament. So the inguinal ligament is this white structure that basically runs oblique across the pelvis, and then once it passes that, this is going to become the femoral artery. The femoral artery is going to be both sides that are labeled here. So once it splits, it's going to become the femoral artery. We also have the deep femoral artery, which is going to be the internal structure as it splits. So we have the femoral artery that's going to be on more of the medial side, and the thinner, smaller structure that's a little bit farther back is the deep. And this supplies supplies the hip joint via medial and lateral circumflex arteries. Together, the femoral artery and the external iliac supply numerous structures in the anterior and posterior thigh. If we were looking at the circumflex arteries, the medial and lateral ones, the circumflex arteries are basically these small structures that run the perpendicular to the bone rather than parallel that we see with the deep femoral and the femoral artery. If we continue to look, now we are focusing on structures on the posterior side, so we see the popliteal artery, and this supplies the knee joint and surrounding muscles. So popliteal basically refers to the back of the knee. So if we look at this diagram here, this is the patella. Basically on the back side of the patella we have the popliteal artery. And this is going to be the posterior view of that here. The popliteal artery divides then into the anterior tibial and posterior tibial artery. So the popliteal artery is going to split off and create a the posterior tibial artery when my mouse is following along here, and the anterior tibial artery that will go then along the front side of the tibia along here. Those will then respectively supply the anterior leg compartment, which includes muscles that are involved with extension and your tibialis anterior, and the posterior compartment, which are involved with plantar flexion and some of your posterior tibialis and flexor muscles. The posterior tibial artery gives off a branch called the fibular artery, and this supplies the lateral compartments of the leg. So if we look here, the posterior tibial, we'll use this picture here. So posterior tibial is going to split off into the fibular artery and supply the lateral fibula of that leg, or the lateral compartment. Remember, the lateral compartment has muscles like fibularis longus and fibularis brevis. So if you don't know which artery supplies blood to these muscles, you do now know. So the fibular artery controls the lateral compartment, the anterior tibial artery controls the anterior compartment, and the posterior tibial artery controls the posterior compartment. The posterior tibial artery then again divides into the plantar artery, medial and lateral. So if we go down to the foot, in my mouse, there we go. The medial side is going to be where your big toe, and the lateral side is going to be for your fifth metatarsal. If we go back to the front, the anterior tibial artery changes its name to the dorsalis pedis artery at the anterior surface of the ankle. So this is actually so it's going to split down here, and that's going to be right near your talocrural joint, it's a little bit distal to that. The dorsalis pedis artery actually has a dorsal pedal pulse that you can take, and it's commonly used when splinting or transporting lower extremity injuries. So if someone fractures their femur or tib fib, so some of the basketball stars, you can look up videos that have basically broken their tibia and fibula and has come through their skin. There's a good video on YouTube if you like to watch those type of things. You have to monitor the blood flow, and one way to do it is through the dorsalis pedis artery, or the dorsalis pedis pulse that sits on the front of the ankle. The plantar arch is then supplied through the dorsalis pedis artery and the lateral plantar artery, and then where they unite to form the plantar arch. And the digital arteries extend from each arch to supply the toes. So we see once that dorsal pedis artery splits off, it then goes underneath with the plantar arch and then individual digital arteries for each of the digits. If we look at the structures for the venous drainage of the leg, the superficial only, these are going to have superficially deoxygenated blood from the foot drains into these. So the great saphenous vein and the small saphenous vein. So the great saphenous is going to be on the medial side, and the small saphenous is going to be on the lateral side. So we have the great is here, and the small saphenous is going to be here. The great saphenous vein drains into the femoral vein, and the small saphenous vein drains into the popliteal vein. When valves become incompetent, varicose veins develop. So when the valves the veins are not able to drain, or the valves are not working in there, basically you get a backup and they become incompetent or unable to function properly. That creates your varicose veins, or the spider veins that you may see on some individuals. Deep to these, we have a few more structures. So we have our plantar posterior sides, and then we have on the plantar and posterior side of the lower leg. The digital veins and deep veins of the foot drain into the medial plantar vein and the lateral plantar vein, and you can see those on the bottom of this image here. And then we also have the posterior tibial vein that runs the backside of the tibia from the popliteal on down to the medial and lateral plantar veins. Deep venous drainage of the leg on the dorsum of the foot or on the top of the foot. The deep veins drain into a pair of anterior tibial veins. So the anterior and posterior tibial veins merge to form the popliteal. So remember, we're kind of working our way up now, right? So we're going to go from the foot, now we're going up the anterior tibial vein, and then into the popliteal or the back of the knee. So remember, this is kind of going to wrap around and run up the back of the knee. So these are basically your condyles of the femur here, and it's going to run in between those. So that's going to be that back of your knee where if someone kicks you, your knee kind of gives out. That's where the popliteal artery and the popliteal vein both run. See some more deep venous drainage of the leg. The popliteal vein becomes the femoral vein, and so that's going to send up from there, from we were at popliteal where my cursor is at, and straight up the femoral vein, and that's going to follow the femur. Okay, so that's going to be that larger structure there. Then remember, the ones on the side of those were the saphenous, the great and the small saphenous that we talked about earlier. Once the femoral vein passes superior to the inguinal ligament, remember that's the white oblique structure that runs right there. Once it passes up past that, that becomes the external iliac vein. So that's where we have our external iliac vein from our common iliac vein, which basically the common iliac vein is going to be the main place that merged to form the inferior vena cava, thus transporting this passage and drainage back to the heart. Neuro, so this is going to be your nerves. First we have a lumbar plexus. A plexus is basically a collection of nerves together. The lumbar plexus is formed from the anterior rami of the spinal nerves L1 through L4. If you have trouble with the rami, anterior, posterior rami, or need to go back to reference spinal nerves L1 through L4, I highly suggest doing that. That was in the nervous system, so under Module 1. The lumbar plexus is divided into the anterior and posterior division. So here's a quick picture, a review of the spinal nerves. The union of the anterior and posterior roots form a spinal nerve. So we have our anterior root here and our posterior root here. That comes together to create a spinal nerve. And the spinal nerves are mixed between motor and sensory functions. The anterior side typically does motor, while the posterior side typically does sensory. Each spinal nerve divides into anterior ventral rami and posterior dorsal ramus. And you can see how that basically the roots come together, create the nerve, split off into rami. The anterior division is we see the main nerve of the anterior division is the obturator nerve. The obturator nerve is going to basically branch off of the anterior side here, and that's going to be this blue structure. So this is going to be on the anterior side, the anterior rami split off, and then they're going to be the orange structures. The posterior divisions are going to be the lighter yellow structure, and the blue will be your anterior divisions. The posterior divisions are the main nerve is the femoral nerve. So the femoral nerve is going to be, femoral nerve is going to be this yellow structure here. This femoral nerve is going to be critical to supplying nervous system to the lower body. We also see some more posterior divisions along the lighter yellow structures. If we look here at the lumbar plexus on a cadaver, we have our ilioinguinal nerve, so that's going to run from your inguinal area and your ilium. We also have your lateral femoral cutaneous nerve, so very superficial femoral nerve. But then we also have our big femoral nerve here that's going to run down underneath this structure and run down. So remember, we talked about some of these structures that run parallel. We have some of our big muscles here that we talked about in class on Tuesday. Basically, these structures are going to run along those nerves the entire length. So if we look at the entire lumbar plexus as a whole, we look at this picture. So we see basically the spinal nerves branching out into the divisions and the rami, and they're going to split off. So we have a ton of nerves that basically split off and are named by the region. So you may want to reference back to Module 1 when we had, it was week 1 or week 2, when we talked about the general anatomy and knowing what areas mean. Ilio hypogastric, when we talked about regions, that's the nerve that supplies that. Ilioinguinal. Genitofemoral, so genitals and femur. Femoral nerve is going to be this big structure here. It's going to run all this structure here. Obturator nerve is going to run through the center of the pelvis and basically run down. And then the saphenous nerve, continuation of the femoral nerve. So it's going to be a specific one that runs off this femoral nerve here and split off into the saphenous. If we specifically look at nerves that make up the lumbar plexus, the first one is the femoral nerve. So the femoral nerve is going to run off of anterior rami L2 through L4. It's going to innervate muscles, such as the quadriceps femoris, which we know is not technically a muscle. It's a group of the 4 muscles together. The iliopsoas, or your hip flexor, and then the sartorius and the pectineus. Cutaneously, basically what the skin sensation for this nerve is going to be the anterior thigh, inferior or medial thigh, the medial side of the leg, and the most medial aspect of the foot. So this green area here is the entire area that if this nerve is injured or severed and basically cut the ability for it to function, this entire green area that you see on this leg model here would lose sensation. The obturator nerve is going to do L2 through L4 as well, but this is going to be on the medial thigh, so your adductors, your gracilis, and your pectineus, and your obturator externus, which is one of your rotators of your thigh. Basically, this is going to be on the medial side here and running just underneath your glutes and run up the medial thigh, superior medial thigh. The sacral plexus is formed from the anterior rami of the spinal nerves L4 through S4. The lumbar and sacral plexuses are sometimes considered together as the lumbosacral plexus. The anterior rami are organized again into anterior and posterior divisions. One of the largest and longest nerves in the sacral plexus and in the body is the sciatic nerve. The sciatic nerve you will hear us talk about over and over again as we continue with the lower extremity. It's composed of 2 divisions wrapped around a common sheath, and that's the tibial division and the common fibular division. So as we see here, this is the basically the back of the leg, and this is going to be the sciatic nerve that basically starts up here near the hip. So this is the top of the hip here. This is the biceps femoris and the semitendinosus. So some of these are hamstring muscles. So the sciatic nerve is going to basically run along that area underneath some of your muscles and split into the tibial nerve and the common peroneal nerve, or the common fibular nerve, and split down. So tibial nerve is going to run through those structures here and innervate your flexor or posterior tibialis and some of your flexors, while your common peroneal nerve is going to split down into your fibularis longus and fibularis brevis, or your peroneal muscle groups. Your sacral plexus is personally a lot easier to trace the flow. So it's going to come off of S4 to L4 and basically split to that sciatic nerve, and that sciatic nerve is such a large, thick structure, and split down into tibial and common fibular nerve. And then after that, it's going to split down a little bit farther into the deep fibular nerve, which is not seen on the anterior view, and then superficial fibular nerve, which is not seen as well. So the superficial fibular nerve wraps around this fibular head here, basically on the front side, and this is kind of how it splits off and wraps around right there. And that's going to be a very sensitive structure if you get hit on the outside fibular head of your leg. Oh, sorry, didn't mean to press the wrong buttons. So the tibial nerve is the first one, and tibial nerve. We see the sciatic nerve is got so many breakdowns of it that we're not going to talk about that specific region. So the tibial nerve itself is going to innervate things like the biceps femoris, and my membranosus, and my tendinosus, so your hamstrings, part of your adductor, flexor digitorum longus, flexor hallucis longus, your gastrocnemius, soleus, popliteus, and tibialis posterior, all things that work on the posterior side of your lower leg. It's also going to do some of your plantar foot muscles, and if you see the branch of the heel and the medial and plantar surfaces of the foot all are innervated cutaneously through the tibial nerve. The common fibular nerve is going to have the short head of the biceps femoris and also some fibular and superficial fibular nerves, and then the fibularis longus and brevis and some of these lower leg muscles. The deep fibular nerve is going to have some of your anterior leg muscles, including your anterior tibialis, extensor hallucis longus, extensor digitorum longus, and fibularis tertius, and the dorsum of your foot, including those same hallucis and digitorum extensors. This is going to do the dorsal interspace between the first and second toes, so basically just in between those two toes, right where my pointer's at, is this fibular nerve going to have a cutaneous innervation. The superficial fibular nerve is going to be your fibularis longus and fibularis brevis, or your lateral leg compartment, and it's going to have a skin innervation on the top of your foot minus that area from the deep fibular nerve, so everything but that area. Alright, so that concludes the PowerPoint over the the nerves of the leg from the hip down. It also included arteries and veins, and I highly suggest you go back to look at your textbook and try to understand the flow and what makes up blood that runs in all these structures and how this oxygenated and deoxygenated blood travels within your body. We will come back and talk about all this with the heart in itself, talk about how blood flow actually works together, and revisit this later on in the semester, but it will be really critical that you understand these structures that you're identifying in lab and connect this back as we continue to understand how the muscles are innervated through the nervous system and how blood functions for your arteries and your veins throughout the lower extremity.